Welcome back to the deep dive. Today, we're really getting into the weeds, maybe the very roots of life itself.
Absolutely. We're looking at how a complex embryo starts to take shape.
Right, that incredible journey from, well, a single cell to something with actual structure. We've got some great sources guiding us.
Sources that really highlight these foundational steps.
Okay, let's unpack this. The core topic is how an embryo develops its basic plan. We're talking germ layers.
The ectoderm, mesoderm, endoderm.
Exactly, those three. And then how the nervous system kicks off with the neural tube.
It's quite the biological ballet.
Our mission, to cut through the jargon, make sense of these complex moves, and give you those, aha moments about how it all starts.
Trying to grasp nature's early engineering, basically.
It's just amazing, isn't it? From one cell, this whole blueprint unfolds.
It really is. And what's captivating looking at the sources is how different life forms solve similar problems in, well, slightly different ways.
Different evolutionary routes to the same destination.
Sort of, yeah. The end result, that basic body plan, seems incredibly conserved, even if the journey varies.
And that's what we're diving into. We'll look at specific examples from the materials. We've got Amphioxus.
The classic primitive chordate.
And amphibians, birds. You'll see the differences, but also the common themes. So let's start with the foundation, getting to that three-layered state. Ectoderm, mesoderm, endoderm.
The absolute building blocks.
Everything comes from these, right? Skin, nerves, muscles, gut.
Precisely. Ectoderm on the outside for skin and nerves, mesoderm in the middle for muscle, bone, blood, and the endoderm lining the inside like your digestive tract.
But how does the embryo actually get these three distinct layers set up? That's the puzzle.
And it's fascinating how different groups manage it. It's not always the same trick.
Let's kick off with Amphioxus. The sources say it starts simpler, just two layers.
Yeah, an outer ectoderm and then an inner layer that's actually a combination of the mesendoderm.
So ectoderm on the outside, mesendoderm lining the inside cavity.
Exactly. Now, to separate that mesoderm out, Amphioxus uses pouching.
Pouching, like little bubbles.
Kind of. Imagine that inner mesendoderm layer, forming outward bulges, pouches, that pinch off.
Okay.
These pouches, which are now the mesoderm, then slide into the space between the ectoderm and what's left of the inner layer, the endoderm.
And they meet up underneath.
They migrate around and fuse on the ventral side, yeah. Then this new mesoderm layer organizes itself.
And the notochord forms here too, from the mesoderm.
Right, from the middle region of that mesoderm, that classic chordate structure, essential for support and signaling later on.
Okay, and the body cavity, the colon.
Also interesting in Amphioxus, in the front part, it forms by that same pouching. The mesoderm pouches become hollow.
Makes sense.
But further back, it's different. A solid block of mesoderm just splits open to create this space.
Two methods in one organism.
Yep, a bit of a mix and match approach to get that internal cavity.
All right, so that's Amphioxus with its pouching. Now amphibians, they start two-layered too, the sources say.
Superficially similar, yes. Outer ectoderm, inner mesendoderm.
But no pouching this time.
No pouching. Here, the mesendoderm just separates. It's more like peeling two layers apart.
Ah, okay. So the mesoderm and endoderm directly split off from each other.
Exactly. And then the mesoderm migrates into position, just like in Amphioxus, but it got there differently.
And the column formation, also different.
Totally different. No pouches splitting open. Instead, the mesoderm sheet itself splits into two layers.
Two layers of mesoderm.
Yeah, an outer one called somatic mesoderm, which snuggles up to the ectoderm.
Okay.
And an inner one, the splanchnic mesoderm, that stays near the endoderm. The space between those two mesoderm layers is the column.
Wow. So splitting the layer creates the cavity.
Precisely. And this mesoderm quickly starts to differentiate into regions, notochord, some mites for muscles, other bits that become kidneys, and so on. Things are already getting specialized.
Okay, moving up the complexity ladder. Birds. And the sources group reptiles and mammals in here too, developmentally speaking.
Right, the pattern for forming these layers is quite similar among them. And it's much more about cell movement, migration.
Less pouching or simple splitting, more choreography?
Definitely. You start with a top layer, the epiblast. Think of it as the source layer.
Right.
Cells destined to become mesoderm and endoderm actually migrate from this epiblast, moving downwards into a space below it called the central cleft space. This space is between the epiblast and a lower layer, the hypoblast.
Okay, wait. So epiblasts and hypoblasts aren't quite ectoderm and endoderm yet.
Good point. No, they're more like temporary positions. The epiblast cells become all three layers eventually. The migrating endoderm cells move into the hypoblast and essentially replace it.
Got it. And the mesoderm cells.
They stay in that space between the epiblast, which will become the ectoderm, and the newly formed endoderm below.
And the notochord in birds, how does that form amidst all this movement?
It's like an assembly line. The cells that will form the notochord gather at the front and then accumulate backwards, anterior to posterior.
Building from front to back.
Yeah, and more cells keep joining from the epiblast to make it longer. It's a very directed construction process.
While all the other mesoderm is also moving into that space.
That's right. The rest of the mesoderm fills in around it.
And then, how does the calum form in birds? Back to splitting.
Yes. Once the mesoderm is in place, it splits into that outer somatic and intersplantic mesoderm, just like in amphibians.
So the migration is complex, but the cavity formation is similar to amphibians.
Exactly. The space between those split layers becomes the calumum.
So, pulling this together for everyone listening, what's the big picture on these 3 layers?
Well, you see these different strategies, right? Pouching in Amphioxus, direct separation and splitting in amphibians, complex migration, then splitting in birds.
Very different approaches.
But the end result, always the same. You get ectoderm, mesoderm, endoderm, and you get a body cavity, the calum.
The fundamental 3-layered plan is achieved, no matter the route.
It's a fantastic example of evolutionary tinkering leading to a conserved, essential structure. Nature finds a way.
Okay, layers established. What's next? Neurulation, making the nerve tube.
Absolutely critical. This is the precursor to the entire central nervous system. Brain, spinal cord. It all starts here.
The very foundations of, well, everything neurological.
Establishing that core axis.
Let's go back to Amphioxus again. How does it handle neurulation? The sources say 3 steps.
Yeah, it's quite distinct. First, the neural plate tissue actually separates itself from the surrounding skin, ectoderm, the prospective epidermis.
It detaches first.
Seems so. Then, step 2, this separated plate folds up, and finally step 3, it forms the tube, and the epidermis fuses over the top.
Why the separation first? Any ideas?
It's thought to involve adhesive changes. Basically, the cells change how sticky they are to their neighbors.
Loosening their grip on the skin cells before folding.
That seems to be the idea, yeah. It's a neat molecular trick.
Now contrast that with amphibians, birds, reptiles, mammals, the vertebrates we looked at. Similar outcome, but different sequence.
Totally different sequence. Here, the neural plate folds first. That's step 1.
Folding before separating.
Right. Step 2, the crests, the raised edges of the fold, meet and fuse together, like a zipper closing.
Okay.
And only then, step 3, does the now closed tube separate from the overlying epidermis and sink inwards.
Wow, so it's fold, fuse, then separate. The exact opposite order for the first 2 steps compared to Amphioxus.
Precisely. And while the tube sinks, the epidermis heals over the top, making a continuous outer layer.
And those neural crest cells we mentioned, they pop off around this time too.
Yes, they sort of delaminate, separate off near the fusion line, and start migrating away to form all sorts of other things. It is a busy time.
So the key difference is when the separation happens before folding in Amphioxus, after folding in fusion in these vertebrates.
That's the main contrast. And just to add another layer of complexity, bony food apparently do it completely differently again.
Huh, of course they do. Nature never makes it too simple, does it?
Never fails to surprise.
All right, so we know what happens, layers form, tube forms, but how? What are the actual forces, the mechanisms driving these shapes? This is where it gets really fascinating, down at the cellular level.
It is. And while we don't have all the answers, we know a lot about the forces, especially in the ectoderm during neurulation. It involves the cell's internal machinery.
We mentioned adhesion changes earlier, cell stickiness.
Critical. Simply changing which cells stick to which and how strongly can drive tissues to separate or fold. It's like microscopic Velcro being turned on and off selectively.
And you mentioned folding. That involves actual contraction, like tiny muscles.
In a way, yes. Cells contain microfilaments, primarily made of actin, just like our muscle cells.
Okay.
Research, like that by Baker and Schroeder in amphibians, showed that bundles of these microfilaments at the periphery of neural plate cells contract.
Pulling the edges.
Exactly. If they're lined up right, their contraction causes the cell to change shape, maybe narrow at one end, and when many cells do this together, the whole tissue bends or folds.
So microfilaments cause contraction and bending. What about other components? Microtubules.
Right, the other part of the cell's cytoskeleton. Microtubules are more like rigid internal scaffolds.
Not for contracting, but for structure.
Primarily. Work by Waddington and Perry, again in amphibians, suggested that the elongation of cytoplasmic microtubules is also key.
How does elongation help folding?
If microtubules align along the main axis of a cell and push outwards, They make the cell longer and skinnier.
Ah, changing the cell's proportions.
Exactly. Making the cells taller and thinner also contributes to the overall folding and shaping of the neural pleat tissue.
So it's a combination. Contraction pulling things in and elongation changing cell shapes.
It seems to be a coordinated effort. Adhesion changes guide separations, microfilaments provide contractile force for bending, and microtubules help elongate cells to facilitate the shape changes. It's quite intricate.
How they all coordinate so perfectly. That's the million-dollar question, I guess.
It's certainly a major focus of research. Understanding that precise coordination is key.
So let's try to wrap this incredible journey up. What's the main thing you, our listeners, should take away from this deep dive?
We've walked through how those first crucial structures form the three germ layers: the body cavity, the neural tube.
And we saw different species use slightly different toolkits, different mechanisms.
Pouching, splitting, migrating.
But they all arrive at the same fundamental result.
Yeah, that's the core message. That three-layered state, the canellum, the nerve tube, this basic body plan is established across the board in these animals. It sets the stage for everything else.
The sources really show how this foundation is laid, getting the embryo ready for the next, even more complex stages of development.
It's a testament to how evolution can converge on successful designs, even via different routes.
And that really brings us to the end of this particular deep dive. Thanks for coming along as we explore these very first steps of life.
It's been fascinating. We've seen the blueprint being drawn. The really exciting question that follows is, okay, you have these layers. How do they then build the actual organs? How does endoderm become a lung or mesoderm become a heart?
Right. What structures come from each layer and how do they form? That's a whole other story.
The next incredible chapter, definitely.
So what stood out to you thinking about these incredibly complex yet coordinated first steps? It's pretty mind-blowing that such different strategies all lead to such similar essential outcomes, isn't it?
Truly remarkable.
We hope this sparked your curiosity. Join us next time for another deep dive.